So the ATI TEAS Science section consists of anatomy and physiology, which is going to go through all the systems of the body and also understanding the different cell types. Then it's also going to cover life sciences, which are going to be about chromosomes, DNA, macromolecules, atoms, periodic table of sequence, physical and chemical characteristics of substances, and acid and bases. The other thing you're going to have to understand, the next section is going to be about scientific reasoning. So this is talking about measurements with like the metric system and understanding data from experiments. And in this video series, we're going to go through all of those things. So I also just want to let you know that I also have an ATI TEAS Science study guide. The link will be below. Go over all of the concepts in this study guide. And if you want the printed out version that you can study from, you can go ahead and order this. So hope you find it helpful. Scientific reasoning portion of the science section of the ATI exam. So we need to know specific measurements using the metric system. So we need to know that there's 1,000 milliliters in a meter, that there's 100 centimeters in a meter, there's 10 decimeters in a meter, 1 meter is 1 meter, 1 decameter is 10 meters, 1 hectometer is 100 meters, and 1 kilometer is 1,000 meters. We need to do the same thing for mass. We need to understand 1 decagram is 10 grams, 1 hectogram is 100 grams, 1 kilogram is 1,000 grams. And same with the liters. Like we need to know 1 kiloliter equals 1,000 liters. So one way to remember this is King Henry doesn't mind drinking cold milk. So it goes kilometer, hectometer, decameter, meter. So that's King Henry doesn't mind. But if you're talking grams, it's not going to be the M, it's going to be the G. Or if you're talking liters, it's going to be the L. But at least this will set it up in the right way. So King Henry doesn't mind. The mind isn't going to be the M, it's just going to be whatever you're talking about, whatever measurement you're talking about. And then drinking cold milk. So deci, centi, milli, okay? Because milli is 1,000, centi is 100th, deci is 1/10, deca is 10 times, hecto is 100 times, and kilo is 1,000 times. So you need to remember how to convert those things. We need to also understand data from experiments. So scientific method is a method of testing an idea by designing and carrying out an experiment and then analyzing the data after. Our hypothesis is the statement or question that we're investigating. A hypothesis is an educated guess on what we think is going to happen. So every study or scientific study has to have an independent variable, a dependent variable, and a control variable. An independent variable is what we think is causing it. So it's something that can be changed throughout the experiment. The independent variable is what we manipulate in the experiment. Our dependent variable is what changes when we manipulate the independent variable. We never manipulate the dependent. That's our results. That's what happens if we manipulate. So we have a plant growing and we want to see if we give it more light, will it grow faster? So our amount of light is our independent variable, and the growth of the plant is the dependent. So we can change the light. We can turn it up higher, turn it down lower on different plants. The dependent variable will be what happens when we manipulate those. Our control variable is what stays constant during the experiment. So that could be the amount of water we give the plants. Every time we grow it, we give it the same amount of water. 3 teaspoons a day or something, or the amount of soil. Like those are our control variables. They have to be the exact same every time we repeat the experiment. And our control group is the group that doesn't receive the intervention. So in that situation, it would be the group that doesn't receive an increased or decreased amount of light, or doesn't receive artificial light, or our scientific experiment will lead to a conclusion. We'll be able to identify, does X equal Y? Does increased light equal increased plant growth? Like whatever we're studying, we can figure out and come to a conclusion. No, it doesn't. Yes, it does. It's the cause and effect relationship. We covered a little bit, but let's go more in depth. So an acid has a pH of less than 7. It ionizes in water. It has a sour taste. Think of like lemons or apple cider vinegar. Turns blue litmus paper red. It reacts with bases and metals, and it donates hydrogen atoms in a solution. Bases have a pH greater than 7. They also ionize in water. They have a bitter taste. They turn red litmus paper blue. They react with acids, and they donate OH negative ions in a solution. pH is measured from 0 to 14. So neutral is 7, and we said below that 7 is an acid, and above that 7 is a base. So a pH of 6 is 10 times more acidic than a pH of 7. And a pH of 5 is 100 times more acidic than a pH of 7. pH of 9 is 100 times more acidic than a pH of 7. So physical properties can be measured without altering the substance. For example, boiling point and freezing point, volume, density, viscosity, all of those things are physical characteristics. We're not changing the substance, so volume measures the amount of space a substance takes up. Viscosity measures the resistance to motion when force is applied. Density is the amount of mass per unit volume. Melting point is the temperature in which a substance changes from a solid to a liquid. Boiling point is when a substance changes from a liquid to a gas. And freezing point is when a substance changes from a liquid to a solid. So when in a solid phase, molecules are really tightly packed together. In a liquid phase, they're relatively close, and in a gas phase, they're further apart, moving all around really fast. Changes in states of matter, like going from a gas to a solid or a solid to a liquid, can be an endothermic reaction, which requires heat, or an exothermic reaction, which takes heat away. So when we change from a solid to a liquid, that change is considered melting, and that's an endothermic reaction. When we change from a liquid to a gas, that's vaporizing, and that is an endothermic reaction. And gas to a liquid is condensing, and that's an exothermic reaction. Liquid to a solid is freezing, and that's an exothermic reaction. Solid to a gas is considered sublimation, and that's an endothermic reaction. And gas to a solid is considered deposition, and that's an exothermic reaction. So there's also something called a triple point, and this is the correct temperature and pressure where all three states of matter can exist. So it can be solid, liquid, and gas, the same temperature and pressure. So chemical properties can only be adjusted by altering the substance. This includes ionization, water reactivity, pH, and heat of combustion. Water reactive substances have reactions to water. There's something called ionization energy, and this is the energy required to remove an electron from an atom or molecule. So enzymes or catalysts are substances that speed up a reaction. We know about this when we talked about pepsin in the digestive process. So they speed up the reaction, but they aren't used in the reaction. They don't get put into the compound. An enzyme lowers the amount of energy that is needed for the chemical reaction to occur. The substance that an enzyme acts upon is called the substrate, and every enzyme has a specific substrate. So amylase breaks down amylose. So solubility is when a substance dissolves in another substance, which the other substance would be considered the solvent. A solvent is able to dissolve. So if you think sugar in water, water would be the solvent. Concentration is the amount of a solute per liter. So the solvent would be water, the solute would be sugar. Osmosis and diffusion. So both are examples of movement at a cellular level, and they're both called passive transport. So osmosis occurs when two solutions of different concentrations are separated by a semi-permeable membrane. So water tends to move across the membrane from an area of low concentration to the area of high concentration, because remember, concentration is the amount of a solute per liter. So think of sugar water or salt water, right? So the salt or the sugar, but let's think salt in this instance. The higher concentration means that it has more salt, and water is what is moving in this passive transport. So it wants homeostasis. It wants it to be equal on both sides. So if this area has a high concentration, has a lot of salt, and this area has a low concentration with no salt, the water is going to move from the area with low salt to the area of high salt, so that the area of high salt will lower until both of them are equal. because it wants to make that equal homeostasis on both sides. So diffusion, so that was osmosis. Osmosis is water movement. Think H2O, right? So water with the O uses osmosis. Now there's something called diffusion. So let's think the same thing. We have an area of low concentration and area of high concentration. Now we still want that homeostasis. Here's our high, here's our low. So what actually happens is the area with the high concentration, the particles that it's high in move, not the water. So if the particles are going to move, they're going to move from the area of high concentration to low concentration because it wants to equal that out. So that's diffusion. So let's talk about the characteristics of water. So water molecule is an oxygen molecule with 2 hydrogens attached to it, and therefore it's very polar. Now, polar means that one side of this molecule, so you have your O with the 2 hydrogen, is negatively charged and one side is positive. Is chemical reactions. So a chemical bond happens between 2 atoms. So chemical reaction happens because of electrons bonding or swapping between 2 atoms. So this bond occurs. So there's 2 types of chemical bonds. We have ionic bonds and covalent bonds. So the difference is with an ionic bond, you have one molecule coming along that has 1 electron on its outer ring. We have another molecule coming along that has 5 electrons on its outer ring. Now, the way atoms work is like swap and share their electrons. So this atom that has 1 electron on its outer ring sees this other atom that has 5 electrons on its outer ring, and it says, hey, you know what? I'm going to give you my electrons so you can be more stable. When it gives this other atom its 1 electron, they bond, right? But this one has no electrons now on its outer ring, and this one is full, and they're both happy, and they've created this bond. They are not sharing the electron. This one has given this other one its electron. That's an ionic bond. Then there's a covalent bond. So in a covalent bond, we have 2 atoms coming along. And let's say they both have 1 electron on their outer ring. They're not going to be like, hey, I'm going to give you my electron, because it just doesn't make sense. One is not more charged than the other. What they're going to do is they're going to share the electrons. They're going to be like, hey, let's be a little more stable, let's share. So they're bonded, but they're sharing the electrons. And the electrons cannot move independently. They're covalently bonded. So in a covalent bond, you have 2 atoms sharing an electron. In an ionic bond, you have 1 atom giving its electron to the other atom, and that is the difference. So in a covalent bond, electrons can be shared equally, and if they're shared equally, it's called a non-polar covalent. When electrons are shared unequally, they form polar covalents. Remember we talked about water being polar? It's because one will be higher charged than the other. So elements also have a charge. So typically metals have a negative charge. So metals gives another atom its electron. This occurs between cations and anions. So a chemical reaction occurs when chemical bonds, so those ionic or covalent bonds, are broken. And there's 5 types of these chemical reactions. So we have a synthesis reaction. This is when 2 or more elements come together to form a single compound. There's a decomposition reaction. So there's 5 types of chemical reactions. There's a synthesis, decomposition, single replacement, double replacement, and combustion. So in combustion, an organic compound combines with oxygen and produces carbon dioxide plus water. In a double replacement reaction, 2 elements switch places with each other. In a single replacement reaction, 1 element switches places with another element. In a decomposition reaction, a single compound decomposes into 2 or more elements or compounds. And in a synthesis reaction, 2 or more elements come together to form a single compound. If you get my study guide, you can see on it. So if you get my study guide, it will show you the synthesis reaction, how it occurs with like compound 1 plus compound 2 equals this new compound AB. So like you can see it much better, and it gives you examples. So that should be helpful too. So all matter in this world is made up of atoms. This table is atoms, I am atoms, these flowers are atoms, my iPad is atoms. All atoms are composed of a central nucleus, which is made of protons and neutrons. So neutrons are neutrally charged. Protons are positively charged. So this nucleus has a positive charge. Floating around the nucleus in various orbitals are electrons, and electrons are negatively charged. So elements, if you think of the periodic table, consists of all different things: hydrogen, oxygen, carbon, all of those things in the periodic table, those are elements. So elements have specific numbers of electrons, mass, and structure. Each element has its own atomic number and atomic mass. The atomic number tells how many protons it has, so how many positive charge that it has. Atoms themselves are neutral, so typically the number of protons that an atom has is also the number of electrons that it has, because the protons are positive and electrons are negative, and we want our atom to be neutral. The atomic mass is the number of protons plus the number of neutrons. So think that center nucleus made of protons and neutrons. The weight of that is the atomic mass. And again, remember the atomic number is just the number of protons. So if we look at the periodic table, so the way it's set up by row is the number of protons. So you have like 1 and then 2's over here. It's 3, 4, kind of skips a bunch, 5, 6. And that's because they're set up into subcategories. So there's like different types of metals and things like that. You don't need to know all that for the T's. There's something called isotopes. And so isotopes are atoms of the same element that have a different number of neutrons. So remember we said that when you have protons, you have to have an equal amount of neutrons. Well, isotopes are things that don't follow those rules. A compound is a substance that is composed of 2 or more molecules. So a compound is something that's composed of 2 or more elements that are bonded together. So you can think of this as like sodium chloride, because it contains 2 elements, sodium and chloride, and they're bonded together. What Mendel did is he took a tall plant that had, think of 2 capital T's. So this is just a tall plant. That's what it is. So it was considered a purebred because both of its parents were tall plants and their parents were tall plants. So this purebred plant, he took it. And then he took a purebred short plant. So that plant's parents were both short, grandparents were short. So he took this short plant and he took this tall plant, purebred. And what that means is the tall one is 2 capital T's, the short one is 2 lowercase t's. And he mated them to make a new plant, considered the F1 generation, because this is the first generation of the mixture of the tall and short plants. All the baby plants came out tall. So why is this? So what this suggests is that all plants have dominant and recessive genes. A dominant gene is the one that's going to show, and a recessive gene is the one you're going to carry, but you don't necessarily show. So the way that this is viewed is something called a Punnett square. And a Punnett square is when we take the gene of one parent, so that 2 capital T's, so it'll be like 2 capital T's. It almost looks like it's like a square, and then it's split into 4 sections. So you have the 2 capital T's down here, and then you're taking the second parent that you had, which was 2 lowercase t's up here. So if you cross them, you have, in your first box, you have the capital T and the lowercase t. In the next box, you have the same thing, capital T, lowercase t, and same for all the boxes. So all of the boxes end up being a capital T and a lowercase t. So what this means is the capital T is the dominant trait. The tall plants are dominant. However, if we take 2 of these F1 generation plants, the capital T and a lowercase t, capital T, lowercase t, they both look tall, but they're carrying the gene for the short plant. And we then meet those 2. What we end up finding is that 3 of the plants are tall and 1 is short. So why is that? We come back to our Punnett square. So we have capital T, lowercase t, capital T, lowercase t. If we cross them, we get 2 capital T's, a capital T and a lowercase t, a capital T and a lowercase t, and 2 lowercase t's. So that 2 lowercase t's, that's the short plant. That plant does not carry the dominant gene. So when we talk about the difference between genotypes and phenotypes, genotypes are the genes we carry. So in that situation, we would have one of the four plants is the genotype, capital T, capital T. Two of the four plants is capital T, lowercase t. And one of the four plants is lowercase t, lowercase t. What this means is one of the four plants only carries the dominant tall gene. Two of the four plants show the dominant tall gene, but also carry the recessive short gene, and one of the plants only has the recessive short gene. When we talk about phenotype, that is what we're seeing. So we're seeing that 3/4 of the plants are tall and 1/4 of the plants are short. Chromosomes, genes, and DNA. So DNA is a nucleic acid. It's located on chromosomes of the cell and in the nucleus. So you might remember this when we talked about the cell going through mitosis and meiosis. Each gene is a section of the chromosome that codes for a protein. DNA is in the form of a double helix, so it's like two strands that are twisted together. And DNA consists of 4 nucleotides. This is adenine, thymine, cytosine, and guanine. So they connect by base pairs, so adenine and thymine pair, and cytosine and guanine pair, so AT and CG. So pretty much in DNA, there's like these two strands that are twisted together in this double helix. And if you zoom in really, really close, there are these nucleotide pairs, and that's how it codes. So if you look at one of the little spindles that are spun together, it'll be like AT, CG, ATAA, TTCCGCG. Like it's just like a code. So both spindles have their own code and they're all twisted together. But C always binds with G and A always binds with T. So then we have RNA. And RNA is when DNA is unzipped and transcribed. RNA is then, because DNA is protected in our nucleus. So RNA will come in and it will transcribe it, and then it will leave the nucleus and go to the ribosomes. Remember we talked about ribosomes, the R ribosomes, they transcribe the RNA. So the RNA leaves the nucleus and goes to the ribosomes. So the thing that is different with RNA than DNA, one of the main things, is that it contains uracil, and uracil replaces the thymine in DNA. So in RNA, the A is paired with U. So the adenine is paired with uracil. So this RNA codes for amino acids that build proteins. So a codon is a trinucleotide sequence. So a codon is a trinucleotide sequence, which just means it's a sequence of 3 of those nucleotides. So remember we talked about the adenine, thymine, cytosine, uracil, or guanine. 3 of those that corresponds to an amino acid in messenger RNA. So mutations happen when there's a mistake. We're going to talk about the life and physical science section of the science section of the TEAS. So the first thing we're going to talk about is macromolecules. So macromolecules are large organic molecules that contain the carbon atom. And in these macromolecules, 4 of the 6 electrons can bond to other atoms. And this is because you know how we have, because you know in the middle of an atom you have protons and neutrons, and then you have the electrons that go around the outside. So the first kind of orbital, you don't have to super worry about this, but the first kind of orbital has 2 electrons, and then outside of that orbital is 4 electrons for these macromolecules. So these macromolecules, they're large molecules and they contain the carbon atom. And carbon atoms are important because 4 of their 6 electrons can bond with other atoms. Most macromolecules are polymers, and what a polymer is a bunch of monomers, which a monomer is a single building block. So it has a bunch of single building blocks to create this polymer that creates a macromolecule. Monomers join together by covalent bonds. Monomers join together by covalent bonds, which then removes water, and this is a process called dehydration or condensation. These bonds can also be broken by adding water, and that's called hydrolysis. So there's 4 types of macromolecules. We have carbohydrates, lipids, proteins, and nucleic acids. So carbohydrates are our source of energy. They are made of carbon, hydrogen, and oxygen. Examples are starches and sugars, and they come from our food sources. So anything that has sugars, starches, or grains, those are going to be our carbohydrates. Our lipids are fat, and it's used for energy storage. And they're fats, and they are our energy source. They also form cell membranes. They also consist of carbon, hydrogen, and oxygen. Example are triglycerides, and in food they're found in fats, oils, and butters. So protein actually works as an enzyme, and an enzyme works to initiate chemical reactions in the body that are needed for life. Structurally, they form muscle and connective tissue, and they're made up of amino acids and carbon, hydrogen, and nitrogen. So an example of this is pepsin, which we talked about was in our stomach to help break down proteins, and it is a digestive enzyme. We also have hemoglobin, which is on our red blood cells, and myosin, which is in our muscle cells. And we get these from leafy vegetables, meats, beans. Then we have nucleic acids, and this is what makes up our DNA, and chromosomes, which codes for all proteins. It's made of carbon, hydrogen, oxygen, nitrogen, and phosphorus, and it makes up our DNA and RNA, and we can get nucleic acids from fish, nuts, and fruit. So there's different types of carbohydrates. There's monosaccharides and disaccharides. So a monosaccharide is the simplest form of a carbohydrate, and it consists of a single molecule of sugar. So monosaccharide, mono usually means one, so it's a single molecule of sugar. A disaccharide, di is two, so a disaccharide consists of two sugar molecules joined together. When this reaction occurs, it actually loses water. When this happens, it actually loses an oxygen. So a monosaccharide, its chemical formula is C6H12O6, where a disaccharide is going to be, because now you have two, so it would be a C12H22 and an O11. And the reason you're missing two H's and an O is because you're losing that water, H2O. Then you have a polysaccharide. This consists of a chain of monosaccharides, and this is starch, cellulose, or glycogen. There's also different types of lipids. Lipids are hydrophobic, meaning they don't like water. They don't attach to water. They're not soluble in water. This is why when you mix oil with water, you can see like the oil and you can see the water. It doesn't. They don't all mix together. So triglycerides consist of fatty acids connected to a glycerol molecule. Examples include fats, so saturated fats or unsaturated fats and oils. There's also phospholipids. So you remember we talked about the cell membrane has phospholipids. It acts as like a barrier for transport, so that's another type of lipid. And there's also steroids, which include cholesterol and hormones such as testosterone and estrogen. Okay, so that's it for macromolecules. So the immune system consists of cells and tissues and organs that work together to protect the body from foreign invaders. This can include infections, antigens, and pathogens. So we have three lines of defense. The first line of defense is a non-specific line of defense that's like our skin or bodily fluids such as mucus, saliva, tears, stomach acid, and they work to keep infections out of the body. The second line of defense is also non-specific, and it attempts to limit the spread of invading microbes. This includes swelling or inflammation at a cut. It signals the body to send white blood cells to this area. It also signals natural killer cells to consume bacteria and to destroy them at the site of the inflammation. Other things that are helpful are phagocytes, which swallow the bacteria, and that has been identified by the helper T cells, and interferon, which combats viruses and blocks cell-to-cell infections. There's also a third line of defense. This is a custom-made defense. It is custom-made to fight off a specific infection. It relies on two types of cells that are made in the bone marrow, and that is the B lymphocytes and the T lymphocytes, also known as B cells and T cells. So B cells produce antibodies to a specific infection. This is known as an antibody-mediated response. They're kind of like memory cells in that they'll remember specific infections. This is what happens when you get a vaccine. Your B cells recognize it and they know there's a problem, And then our T cells, which are made in their bone marrow but mature in our thymus gland, they attack the pathogens. They're also known as killer T cells, and they pretty much move about the body looking to recognize these pathogens that they've seen before. Both helper T cells and B cells recognize infection. Physiology of the urinary system. So this consists of organs that excrete and eliminate waste from the body. This includes our kidneys, our uterus, our bladder, and our urethra. Toxins are taken out of the blood by the liver. The blood then goes to our renal arteries in our kidneys. So the filtering of waste first starts in our kidneys. Kidneys are known to be the key organ in excretion. So the liver kind of takes the waste out of our blood, but the kidneys has to do with excreting of the waste. Each kidney is divided into two major parts, the medulla and the cortex. So each kidney has about 1 million filtering tubules or nephrons, which perform three processes: filtration, secretion, and reabsorption. Each nephron tube has a glomerulus. This is a cluster of capillaries that act as a filter, and they keep key things in the body and take things that they want out, and which then helps to excrete those things that they don't want, the waste, out through urine. And the kidneys are key in providing homeostasis for our body. It's going to be about the organ system within the body. So this first section we're covering anatomy and physiology. So let's go through. So all cells in the body have specialized functions, and they join together to make tissues. There's different types of tissues in the body. There's 4 types of tissues in the body. So we have connective tissue in our bones and cartilage, muscle tissue in our cardiac and skeletal muscles. We have nervous tissues in our brain and spinal nerves. We have epithelial tissues, which are inside our mouth, on our skin, and on organ surfaces. So these tissues join together to make organs in the body. And all of the organs in the body work together to maintain homeostasis, because they want this kind of equilibrium in the body to keep everything, healthy and in line. And that is when we're healthy. And when our body isn't in homeostasis, that's when we're sick. So there are 10 organ systems in the body. We have our respiratory system, our cardiac system, our gastrointestinal system. We have our neuromuscular system, reproductive system, integumentary system, endocrine system, our urinary system, and our immune system. So we're going to go through all these systems in this video series. So something else that we need to know for the ATI TEAS is anatomical planes of the human body. A coronal plane divides the body into 2 parts. It goes through like this, okay? So think if you had like a square through. So it's separating my body into the front and into the back. That's the coronal plane. Another word for that would be the anterior and posterior, or the ventral and dorsal. So dorsal's the back, ventral's the front. Then there's also the sagittal plane. Sagittal is going to divide the body like this into the right and left sides. And we have a transverse plane. This divides the body into the top and the bottom. So the transverse is coming through like this way. So we have the top and the bottom. So one more time, the coronal plane is cutting us this way. So we have the front, or the anterior, or the ventral. And then we have the back, which is the posterior or the dorsal. Okay, then we have the sagittal plane that divides us this way into the right and the left, and the transverse plane that divides us into the top and the bottom. This is the theory that all living things are composed of cells. All cells come from pre-existing cells. They do this through mitosis and meiosis. They carry out basic processes. So some cells are used for food and metabolism to make our food into energy, and then other cells are responding to the environment, and other cells are growing and reproducing and disposing of waste. There are 2 main types of cells. You have prokaryote cells and eukaryote cells. So in a prokaryote cell, it sort of looks like this. It has what are called pili, which are like little attachments. They're like little fingers that come off of it. It also has these long little tails at the end, and those are the flagella. It has a plasma membrane. It also has a capsule with a sticky coating and a cell wall that provides rigidity. So in the middle, you have a nucleoid, which has single cell bacterial chromosomes. And then you have ribosomes all in there as well. So you won't need to know what is in each cell by heart, but you will need to know what things within the cell does. So we'll get to that. So a eukaryote cell, so we just went over a prokaryote. Those are the ones, they look like this. They have little fingerlings around it, and they have the flagella tail at the end. Then you have a eukaryote cell. That's going to be a circular cell that has a plasma membrane. And within the cell, there are multiple different things. It's not as simple as the prokaryote cell. So you have ribosomes, a nucleus, centrioles, microtubules, ribosomes, cytoplasm. There's a Golgi apparatus, and all of these things do different things within the eukaryote cell. It has a smooth endoplasmic reticulum and a rough endoplasmic reticulum. So we'll go over all of this stuff, don't worry. So what are the difference between the prokaryotes and the eukaryotes? We just kind of went over a little bit of that. So prokaryotes, they're simpler. They're single-celled. They're usually bacteria or algae. They're very small. They have no nucleus or organelles. And they have single, usually circular chromosomes, and they reproduce by budding or fission. So they just kind of separate. Eukaryotes, on the other hand, can be singular-celled or multicellular. They're plants, cells, animal cells, fungi. They're large cells. They're 10 times larger than a prokaryote cell. They have a nucleus and they have organelles. They have multiple chromosomes. They separate by mitosis or meiosis. So if we kind of break that all down super simply, the prokaryotes, they are single-celled. They're really simple. They're small. They're bacteria. They're fungi. The eukaryote cells, that's where you're getting into the plant animal cells. They have multiple different things inside of it that do multiple different things, which we'll go over. And they use mitosis and meiosis to reproduce. Okay. So what are the functions of these organelles? So you have a chloroplast. This is the site of photosynthesis, and this is in eukaryote plant cells. So if we think like plants, we think of chlorophyll. So plants have chlorophyll, so like a chloroplast, and this is what it uses for photosynthesis, okay? All eukaryotes have a nucleus. This regulates all cell activity, including replication of DNA, which codes for enzymes that carry out all important cell jobs. So we always think of the nucleus as like the brain of the cell, I guess, right? So that's the nucleus houses the DNA. And so this nucleus has the DNA inside of it, and our DNA codes for certain enzymes that carry out all important jobs within the cell. So that's why the nucleus is known as like the brain of the cell. Then we have ribosomes. Ribosomes are in eukaryotes and prokaryotes, and ribosomes use RNA to transcribe DNA into protein. So ribosomes use RNA to transcribe DNA into proteins. So think a ribosome starts with an R. So ribosomes use RNA to transcribe the DNA into proteins, okay? Then we have the mitochondria. This is the powerhouse of the cell. This uses oxygen to burn glucose and produces ATP for cell energy. So our mitochondria is where our energy is created. A mitochondria is in all eukaryote cells. So plant and animal cells, this is like the little place that's creating the energy. Mitochondria. The cytoplasm is a watery like substance that's inside the cell. And cytoplasm is like this medium that holds all the little organelles in the cell into place. Cytoskeleton is what provides structure for the cell, and a cytoskeleton is only in a eukaryote cell. A cytoplasm is in both a prokaryote and a eukaryote. So in endoplasmic reticulum, there's two types. We have a smooth and a rough. The rough, again, think the R, has ribosomes in it. And the rough endoplasmic reticulum with the ribosomes produces proteins. The smooth endoplasmic reticulum is used in the synthesis of fat. So again, R in the rough endoplasmic reticulum has the ribosomes, and the smooth endoplasmic reticulum with the S synthesizes fats. There may be a test question on that on the exam. So the cell membrane is a phospholipid bilayer, and what this means is there's like a little circle with a tail one way, and then a little circle with a tail the other way. So it's a bilayer. So there's two phospholipids. Those are the phospholipids, and they surround the cell. And this is a highly selective barrier for passive and active transport. So this phospholipid bilayer works to allow things into the cell and out of the cell by different mechanisms of transport. There can be, certain little things where a protein fits like a lock and a key that opens up the bilayer to let that protein in. It can also be things like diffusion. You'll see that a lot when you get into nursing school with electrolyte imbalances and things like that and fluid overload. And are we giving hypertonic solution or hypotonic solution? All of those things, again, come back to this phospholipid bilayer. And this is why this information is really important to know before you go into nursing school, because it all carries through into the medicine that we learn. Okay, let's keep going. There's also a cell wall. This is a stiff outer layer of the cell, and this is in plants, fungi, and prokaryotes. So Golgi bodies are something else that are in eukaryotes. These package proteins and secrete them outside of the body. So these are like little guys that package in the proteins, and then they go to there and they let it out. Vacuoles, these are like storage containers within the cell. So again, we can think of vacuole. It's kind of like a vacuum, and the vacuum like stores things, like kind of like sucks them up and stores it. So think of it like that. And flagellum, remember we were talking about the prokaryotes have that long tail, and that's the flagella. That is used for locomotion, and this can be both in prokaryotes and eukaryotes. Is cellular reproduction. So mitosis versus meiosis. Both of these are ways that eukaryote cells reproduce. But what is the difference? Now, you don't need to know this like perfectly. You don't need to know every single step, but you do need to know the gist of it. So let's go through it. So mitosis occurs when cells split. So mitosis happens when a cell duplicates itself. It can happen in a single cell organism like a protozoa or a bacteria, or it can also happen with other living things when they grow or heal or reproduce. These new cells are identical copies, so they have the same DNA as the first cell. So what are the steps of meiosis? So first you have prophase. In prophase, the nuclear membrane around the cell starts to dissolve. This allows the double chromosomes, which look like X's, to float freely amongst the cell. Spindle fibers, which are like these little spindle fibers, start to conjugate around the structure, and they produce a spindle apparatus. So they kind of connect to these little chromosomes, and then they're like connected to the end of the cell. And then this will separate the DNA into two separate poles. During metaphase, this is when it starts to pull the DNA towards opposite ends of the cell. And then during anaphase, they start to pull apart these DNA X's into two, they're like V's. So it kind of like starts like this, and then it gets pulled away in two V's like that. And then telophase is when this process starts to reverse. So the spindle fibers dissolve, the nuclear membrane starts to come back around to create two separate cells, and these two cells then go through cytokinesis, and this is again where the two cells are divided by a cell membrane. So now you have two daughter cells. So again, steps of mitosis. We're going through prophase, metaphase, anaphase, telophase, and then cytokinesis. So things that are important to remember. You have those little X's of DNA. The prophase comes in. It starts to let those X's move freely. Then you have metaphase, where it starts to pull those X's that are DNA, pull them towards the opposite ends of the cell. The anaphase pulls them apart completely. They're now two separate little pieces of that DNA chromosome. And then telophase, it starts to reverse. You start getting the nucleus around the cells, and the cytokinesis is when they are completely divided and there's a new nucleus within that cell, and you now have two identical daughter cells. Okay, so now let's talk about meiosis. This is more complex than mitosis. It has two parts. There's meiosis 1 and meiosis 2. However, if you understand mitosis, it's very, very similar. So meiosis 1, there's meiosis 2. That's because it goes through two phases of prophase, two phases of metaphase, two phases of anaphase, and two phases of telophase. Prophase 1 is similar to the mitosis prophase. This is, again, that nuclear membrane starts to disappear. Chromosomes from each parent cell are then able to kind of mingle. During this is when the chromosomes cross over. So a chromosome from each parent bundle together. So you know you have like 2 chromosomes in the X. So you're getting one from one parent, one from the other. And this is what causes genetic variation. When you know you're a child with a parent, this is why there's genetic variation, because each parent comes in with 2 chromosomes and then they swap them, but those chromosomes are not identical. So that's where we get the genetic variation. So then we have metaphase 1, anaphase 1, and telophase 1, which mimic the mitosis. It's the same thing. So if you understand mitosis 1, then you understand meiosis 1. After meiosis 1, we then have that cytokinesis again, where it's separating into 2 separate cells, and it's creating these 2 cells, and they're called 2 diploid cells, where they contain full, unique DNA. It has the 46 chromosomes, and again, it's a mixture of those parent cells, and that's why we get the genetic variation. So these 2 diploid cells then start to go through meiosis 2. So we have prophase 2, metaphase 2, anaphase 2, telophase 2, and they mirror the similar process of mitosis. We have those centrosomes and spindle fibers that push apart the chromosomes. And when those spindle fibers retract, the chromosomes separate, and now you have 23 chromosomes in each cell, not the 46. So this results in 4 haploid cells, or gametes, that have 23 chromosomes. Meiosis 1 is giving us 46 chromosomes in 2 daughter cells. And then meiosis 2 is then separating those 2 daughter cells that each have 46 chromosomes into 4 haploid cells that each have 23 chromosomes. Anatomy and physiology of the respiratory system. So the respiratory system, its function is to take in oxygen, to oxygenate our blood and our organs, and release carbon dioxide. This oxygen provides energy for the cells in our body, and this is accomplished through gas exchange. So the gas exchange process, this is automatic. When you're breathing, you're not thinking about it, you just automatically breathe. The air comes through your nasal cavity into the pharynx, which is the area like in the back of your throat, down through the larynx, which is your neck, and the larynx is where your vocal cord is. Then it comes down through your trachea. In your trachea, there's something called the epiglottis. This is a flap that solid food and liquid don't go into your lungs. So this epiglottis kind of protects the lungs when you're eating or drinking. So the trachea branches off into 2 bronchi into the lungs, which then branch out into narrower sections called bronchioles. So it goes even narrower into bronchioles. And then at the end of the bronchioles are little sacs called alveoli, and they look like little grapes. They're just like little circles. When a person inhales, these alveoli inflate and then deflate. They're like little balloons on the ends. The alveoli are the main site of gas exchange. Important to know that. The gas exchange process takes place between the alveoli and the blood. In these alveoli, there's surfactant, and that allows the alveoli to inflate by lowering surface tension. The surfactant reduces the pressure required to inflate the alveoli. And then oxygen passes through the alveoli wall into surrounding blood capillaries. So there's like these little balloons at the end, the alveoli, and there's little blood capillaries all around them. And when it passes through those capillaries, the oxygen is then grabbed by the red blood cells. When a person exhales, this whole process is reversed. Carbon dioxide goes into the alveoli, up through the bronchioles, up through the bronchi, up through the trachea, up through the larynx, up through the pharynx, and then out your nose through your mouth. Your diaphragm flattens to bring air in, and it expands to push the air out. Okay, and that's it for the respiratory system. We're going to be talking about the anatomy and physiology that you need to know for the ATI TEAS Science portion of the cardiovascular system. The function of the cardiovascular system is to transport materials to and from different cells in the body. Blood is the major carrier of these materials. They include nutrients from the digestive system, oxygen from the respiratory system, hormones such as insulin, which are secreted by glands in the nervous system, and immune cells that help to fight infections. Things that the cardiovascular system carries away are waste products that eventually get carried out through the urine, carbon dioxide that is exhaled, and excess salts and other materials that are retained in the body. So the cardiovascular system, it's like the transportation system throughout the body, moving waste and nutrients and oxygen throughout the entire body. We need to understand the difference between an artery and a vein. So an artery has thick walls, and they carry oxygenated blood away from the heart. So the way I remember this is the arteries, since they carry blood away, so the A, right? A and A. So arteries carry blood away from the heart, and they're thicker because that heart is pushing that blood super hard, and those are the first things that are getting that blood. So they need to be stronger. They need to be thicker. So that's just the way I remember it. Then we have veins. Veins are thinner, and veins carry deoxygenated blood from the cells of the body back into the heart. So if we think the heart is the one that pumps the blood throughout the body. So those arteries are going to be thicker, right? And they're carrying the blood away from the heart. So they need to be able to take the pressure of how much pressure that heart is pushing. But as it goes through the body and loops back around, it's coming back into the heart, it comes through a vein. And those can be thinner because they're not getting that immediate pump, right? They're getting kind of the tail end of it. So they're just thinner, and they're carrying the blood back into the heart. They're just kind of pouring it into it. So the human heart has 4 chambers. We have the right atrium and the right ventricle, and the left atrium and the left ventricle. So blood is carried from the body through the veins into the right atrium, so that top part, through the right atrium from the upper and lower body. In that right atrium, it then goes through the tricuspid valve, which kind of it will open and let the blood into the right ventricle, and then it closes so that blood can then move into the lungs. And then as the blood moves into the lungs, that tricuspid valve will then open and refill it. So the blood's coming in from the body through the veins into the right atrium, through the tricuspid valve into the right ventricle. From the right ventricle, it then goes into the lungs. So that right ventricle doesn't need to be super, super strong, right? Because it's only making the blood go from the bottom of the heart into the lungs. It's only moving that far, not too far. So from the lungs, it then goes into the left atrium. From the left atrium, it goes through the mitral valve into the left ventricle. Now, the left ventricle is the thicker ventricle. It has more muscle. It's stronger because from that left ventricle, that blood needs to be pumped up through the entire body with enough force that can get it through those arteries, through those veins, and back into the heart. So the blood from the left ventricle is then pumped up through the aorta and throughout the entire body. And that blood is oxygenated, right? Because it went from that right ventricle to the lungs, got oxygenated into the left atrium, through the mitral valve, into the left ventricle, shoots up through the aorta and into the body to oxygenate the entire body. So in the body, there's 2 loops. We have the pulmonary loop and the systemic loop. So we actually just talked about this, but let's just define it. So the pulmonary loop is taking that deoxygenated blood from the right ventricle, pushing it through the lungs and getting it oxygenated into the left atrium. Okay, so that's the pulmonary loop. It's the way that the heart connects to the lungs to oxygenate the blood from the right ventricle through the lungs into the left atrium. We then have the systemic loop, and that's the loop that is going from the left ventricle, pushing that blood up through the aorta and through the entire body, back through the veins and back into the heart. The heart moves on a rhythmic contraction. This is our heartbeat. So we have systole, which is when the heart contracts, and then diastole is when it's relaxing. In the heart, there's also something called the sinoatrial node, which is also called the SA node, and this is the pacemaker of our heart. It's located in the upper right atrium, and it controls the heart muscle contractions by electrical activity. So this SA node is a pacemaker of our heart located in the upper right atrium, and it controls the contraction of our heart through electrical signals. Okay, the last part of the cardiac system, we're going to talk about components of the blood. So the things that are in our blood that are carried throughout our body by the cardiac system. So we have plasma. This is the liquid component in our blood that just helps to keep things moving through the system. We have erythrocytes, which are also called red blood cells, and these are what attach to oxygen and keep our cells oxygenated throughout our body. The oxygen is carried on a protein called hemoglobin, and it's carried our lungs throughout our body, and then attaches to the carbon dioxide and carries that back to our lungs to be released. We have leukocytes, which are white blood cells. These are made of 5 types that help to fight our infections in our body. So we have white blood cells, which are leukocytes, and these attack and destroy harmful things in our body. Platelets are fragments of cells that are used to clot in our body. So if we get a cut and that needs to clot, so we stop bleeding, those are platelets that are used. And then we also have the lymphatic system. This is a network of capillaries and veins that carry interstitial fluid and waste through the body called lymph. In this video, we're going to be talking about the central nervous system. So the central nervous system includes the brain and the spinal cord. It also is made of neurons, which are basic nerve cells that conduct electrical impulses. They conduct this information electronically along a axon fiber, which is a long extension of the nerve cell body. So the nerve cell body kind of looks like there is this, almost like a circle nerve cell with a nucleus, and then it has this axon body coming off, and then it has an axon terminal at the end, which are like little fingers sticking out. So information is sent along this axon body, is then sent out through the axon terminal, and then it's collected on the next nerve cell on the dendrites, which the nerve cells here with the nucleus, and it has little dendrites that come off of it. So you have the nerve cell with a nucleus and the dendrites with the axon tail and the axon terminal at the end. So the electrical impulse or electrical information comes in through the dendrites, through the nerve cell, along that axon, and then out through the axon terminal. Information is sent both electrically and chemically. If it's sent chemically, it's sent by something called neurotransmitters. So these little chemicals, what are called neurotransmitters, then stimulate that nerve, okay? You can think about that with medication or dopamine or things like that. So we're also going to talk about the peripheral nervous system. So the peripheral nervous system, you think, right? So central, brain, and spinal cord. Peripheral is outside of that. So it connects through the central nervous system. It consists of somatic, which are voluntary nerves, and automatic or involuntary nerves. So somatic movement occurs by an electrical signal sent from the brain to motor neurons in the body. So with somatic movement, it's movement that is voluntary. So I'm going to lift my hand up. That's somatic. So what is happening? So my brain is sending a signal to the motor neurons. The motor neurons are then sending the signal to my muscles saying, hey, I want you to lift that hand up. My hand gets lifted up through a neurotransmitter called acetylcholine. And acetylcholine is what is used to make my muscle contract or lift. So for this next portion, we're going to be talking about the brain. So different portions of the brain have different responsibilities and functions. Now, I love the brain, so I'm excited for this one. Okay, so we have our brain is composed into different lobes. So we have the frontal lobe in the front. We have the occipital lobe in the back. Then we have the parietal lobe, which is on top. And then our temporal lobe is kind of underneath in the middle, and our cerebellum is in the back at the bottom. And then we have the brainstem. Okay, so let's just start there. So frontal lobe. So our frontal lobe has to do with our movement, our reasoning, our personality, our behaviors, and our memories. So a lot of patients that might have trauma or injury to their frontal lobe, we see this a lot in psychiatry. They have a lot of like impulse control or behavioral issues can occur from that region. So the thing that's cool with the brain is when you have an injury to the brain, if you can locate where that injury is, you can understand certain behaviors or illnesses. Like if people are having trouble seeing, it could be damage to the occipital lobe. Like it's just so cool. Okay, sorry. So let's go on to the parietal lobe. So that's the one kind of in the top back. So the parietal lobe, this is our intelligence, our reasoning, telling right from left. Our language comes from the parietal lobe, sensation and reasoning. Our occipital lobe is in the back, and that's our vision. Our cerebellum is below that. So like the occipital cerebellum is below. That's going to be our balance, coordination, and fine motor control. And then we have our temporal lobe. That's one kind of like in the middle. This is our speech, behavior, memory, hearing, and emotions. And then our brainstem. Our brainstem controls our breathing, our blood pressure, our heartbeat, and our swallowing. So our frontal cortex really performs the most sophisticated functions of the brain. Our sensory and motor cortex, that is what we were talking about with motor function, with our central nervous system. So the motor function and the sensory system. So our sensory cortex and our motor cortex are very close together. Our frontal lobe does our planning, reasoning, imagining. So our frontal lobe is where decision making largely occurs, and that's why we do see the impulse control problems with people that do have injuries to their frontal lobe. And again, our brainstem controls our breathing, our swallowing, and our blood pressure. So if you see patients with an injury to their brainstem, you've got to be watching for that breathing and the blood pressure. That's why sometimes like people snap their necks and they die. That's what it is. You're not breathing. You're not having good blood pressure. Brainstem. All right. And that is it for the brain. Let's talk about the muscular system. The muscular system is used to maintain posture and create movement in the body. There are three kinds of muscle tissue. We have skeletal muscle, smooth muscles, and cardiac muscles. So cardiac muscles are the muscles of our heart. Our skeletal muscles attach to bones, and our skeletal muscles are the only ones we consistently can control. And smooth muscles are those around our organ. They are in our digestive system, and they have involuntary movement. So something to know about muscles is they can only contract. They can't extend. So our movement is created through muscle contractions and relaxing. So, right, because if like my arms are out, I can contract it or I can relax it. So muscles are arranged in antagonistic pairs. So what that means is like you have a bicep and a tricep. So when the bicep contracts, the tricep has to relax. And when the bicep relaxes, the tricep is contracting. So bones are connected to structures through tendons and ligaments. So tendons connect bones and muscle, and ligaments connect bones to bones. So typically a ligament is like a joint. So let's talk about the skeletal system. So our skeletal system is made up of 206 bones in the body. You have different types of bones. There's long bones, there's short bones, there's flat bones, and there's irregular bones. But bones aren't just the skeletal system in our body. So bones consist of two types of tissues. We have compact bones, which is a dense hard tissue that forms the outer layer of most bones in the shaft. Inside of that, there's nerves and blood vessels. Then the spongy bone, that's found on like the ends of the bone, and those kind of, they call it sponge, kind of looks like sponge, like these irregular sheets that make up the inner part of the bone, because on the outside is the compact hard, and then you get into those spongy sheets. And they're only located on the ends of the bones. In some bones, like our pelvic bones, our skull, our ribs, and vertebrae, it is found in the center, but when you're thinking of like a regular bone, the spongy part is just found on the inside of the outer portion. So the spongy bones on the inside outer portion. So in the center, so we have, if you think of a regular bone, the center, this is the compact, hard, dense bone, makes up the outside and around the whole bone, right? And then inside of that, there's nerves. But then on the ends, you have the spongy bone, and inside of that, you have red and yellow bone marrow. So The red bone marrow consists of blood stem cells and blood cells in various stages of development. That's where they develop. So you think the red bone marrow is associated with red blood cells. And it creates most of the body's blood cells, not just the red blood cells. It also creates white blood cells and platelets, and it helps to eliminate old blood cells. So the blood cells are being created in that red bone marrow. The yellow bone marrow is found at the center of long bones, and it consists mostly of fat. Anatomy and physiology of the reproductive system. So reproductive systems obviously consist of two different systems. You have the male and you have the female. So the female reproductive system consists of ovaries. So you have two ovaries, and in those ovaries you have a bunch of eggs, which are also called ova. So on that, there's some tubes called the fallopian tubes, and that's where when the egg is dropped to go down to the ovary, it travels through the fallopian tubes and heads down to the uterus, and the uterus is also known as the womb. This is where an embryo develops to be born. When the sperm comes into the uterus and fertilizes the egg, it then attaches to the endometrium of the uterus. So it attaches to the wall, and that's where the baby can grow. The actual vagina itself is just a passageway, essentially, for the baby to come out into the world. So you have your uterus, and at the bottom of the uterus is the cervix. It's like just like this little hole. It's like little teeny tiny hole. But after the baby starts growing and different chemicals throughout your body, that cervix begins to expand. It opens up to about 10 centimeters, which is pretty big. And then that baby can just come right out into the world. The male reproductive system, on the other hand, consists of two testicles, or testes. So sperm production is created here in the semiferal tubules. The epididymis is like the storage area for the sperm. Then there's something called seminal vesicles, and this is something that secretes mucus and fructose and the hormone prostaglandin that mixes in with the sperm. And then there's the prostate gland, which is a large gland that secretes semen. All right, so let's talk about sexual reproduction. So sperm made in the testes, and it's controlled by the hormone testosterone. Eggs are produced in the ovaries, and once it's mature, it can enter into the fallopian tube. Fertilization occurs when one sperm meets one egg. So the egg and the sperm meet, and then it moves into the uterus, where it will then try to attach to the endometrium, where then it can grow into a baby. If it fails to be fertilized, the egg will eventually dissolve, and then a woman will menstruate. So this system is also connected to the endocrine system, because the endocrine system during this entire process is releasing hormones that regulate most reproductive systems in our body. This is through hormones like estrogen, testosterone, and progesterone. And physiology of the endocrine system. So the endocrine system produces hormones for our entire body. These hormones control growth, control sexual development, control metabolism. These hormones are secreted from glands into our bloodstream and transported throughout our entire body. An example is the hormone insulin is made in our pancreas, and then it is transferred, and it helps to regulate glucose in our blood from carbohydrates. Things to know, so growth hormone, that is created in the hypothalamus and the pituitary, and that functions by growth, like helping us grow. Oxytocin and vasopressin, these are made in the hypothalamus, and this helps uterine contractions. We then have thyroxine, which is made in our thyroid gland, and that helps our metabolism. We have insulin and glucagon made in our pancreas that helps to regulate blood sugar. We have cortisol that is made in our adrenal cortex, and that helps with stress and metabolism. And we have estrogen and testosterone made in our ovaries or testes, and that helps with sexual reproduction. Of the integumentary system. So this consists of our skin, our hair, our nails, our glands, and our nerves. Skin is the outer covering of our body, and it's the largest organ. Our skin is actually an organ, I know, it's pretty crazy. It makes a barrier that protects our body from physical damage, ultraviolet heat, chemicals, and diseases. So the epidermis is the outer layer. 90% of it consists of cells called keratinocytes. The protein keratin makes these cells tough, scaly, and resistant to water. 10% of epidermal cells are melanocytes, and these produce the pigment melanin. And this protects us from ultraviolet rays and sunburn. So under the epidermis, then we then have the dermis. It's the middle layer. It's dense, it's thicker, and it's connective tissue, nervous tissue, and blood vessels. And then right below that is a thin layer of hypodermis, which is the inner layer of our skin, and it connects to muscles underneath and bones, and this is the subcutaneous tissue. So glands in the integumentary system include exocrine glands that produce sweat, glands that secrete product through ducts. So the pseudopharius gland, it's also known as the sweat gland, secretes water and sodium chloride, and it serves to lower body temperature. Sebaceous glands produce sebum, or an oil secretion that lubricates the skin and makes it more elastic. And then ceruminous glands are found in our ear canal, and they secrete ear wax, which is called cerumen. And it protects the ear canal and it lubricates it. So hair is also an organ that consists of dead keratinocytes. Structurally, it has 3 parts: the follicle, the root, and the shaft. Hair is found in most areas of the body, and it protects us from UV radiation and insulates our body from the cold. And our nails are also organs found at the distal end of our fingers and toes, and they're hardened keratinocytes, and they protect our fingers and are useful for scraping or scratching. Talking about the anatomy and physiology of the gastrointestinal system. So the function of the gastrointestinal system is to break down food both physically and chemically. Digestion occurs as the breakdown of food into its molecular particles. These essential nutrients from food are absorbed through our digestive tract into our bloodstream and move throughout our body as nutrients. So the gastrointestinal tract runs from our mouth all the way down to our butt. In between, we have our esophagus, our stomach, our small intestines, our large intestines, and those are all connected into this gastrointestinal system. So digestion begins when we start to chew our food. Mechanically chewing of our food creates saliva and is what starts the digestive process. Saliva moistens our food as we begin to digest it. The food then gets shaped into what's called a bolus. So if you see that on your exam, that is just like, think of chewed up food. That's a bolus of food, or a ball. And we swallow it. And it goes down, like we talked about with the respiratory one, it goes down our pharynx, larynx, that epiglottis, make sure that it doesn't go into our lungs, passes into the esophagus, right? So not the trachea. That epiglottis is at the top of the trachea, making sure it doesn't go into our lungs. So that's where it splits into the trachea, esophagus. So it goes into our esophagus next and down into our stomach. And in our stomach, we have gastric juices that consist of hydrochloric acid and has a pH of 1 to 2. So in one of the videos, we're going to talk about pH, but what that means is it's highly acidic, so it can break down that food. The stomach also uses an enzyme called pepsin to break down food. So the gastric juices serve 2 purposes. One is to break down food, and one is to kill bacteria in the food. The pepsin works to digest proteins. The food mixes with water and creates this like creamy substance, which is called chyme. The food moves from our stomach throughout our system through mechanical contraction known as peristalsis. So that's kind of like this mechanical contraction. It's automatic, that's just pushing the food throughout our body. So in the stomach, the food is going to pass through something called the pyloric sphincter. And this pyloric sphincter is right at the bottom of our stomach. And it's when it passes through there, it then goes into our duodenum, which is the first part of the small intestines. Here, proteins are broken down into amino acids and starches are broken down into simple sugars. So when it breaks down into these simple things like amino acids or simple sugars, it can then be absorbed into the body through the wall of the small intestines and can be carried as nutrients to our cells. So any undigested food stays in the small intestines and then passes into the large intestines and then exits our body as waste. Okay? And that is it. So let's do a quick review, okay? So gastrointestinal system starts in our mouth, out through our bottom. We eat food, we have the saliva. The saliva helps to start the digestive process. It turns that food into a ball or a bolus that we then swallow through our pharynx, larynx. It then splits at the epiglottis. The air, remember, goes into the trachea. The food goes into the esophagus, down into our stomach where it's highly acidic and has acids and pepsin that break down the protein. So break down that food, goes through the pyloric sphincter into the duodenum. More proteins are broken down into amino acids, and carbohydrates are broken down into simple sugars. It's then absorbed through the small intestinal wall. The waste then moves through the small intestines, through the large intestines, and out our body. And that is the summary of the gastrointestinal system. This video a thumbs up. Make sure to subscribe below. If you want that free 14-page ATI study guide, make sure to grab one down below. If you want the full ATI TEAS Science study guide, you can go ahead and click that link below as well, and it will bring you over to where you can purchase. Okay, thanks guys.